From the JAMA Network, this is the JAMA Medical News Podcast, discussing timely topics in clinical medicine, biomedical sciences, public health, and health policy, featured in the medical news section of JAMA.
I'm Rita Rubin, lead senior staff writer for JAMA Medical News. Today we're joined by JAMA Associate Editor, Dr. Gil Rabinovici. Dr. Rabinovici is a neurologist and director of the University of California, San Francisco Alzheimer Disease Research Center. We're going to be discussing highlights from the recent annual Alzheimer's Association International Conference, or AAIC for short, in London, which he attended. Welcome, Gil.
Thank you so much, Rita. It's really a pleasure to be here and to summarize what I think was a really exciting conference with a lot of momentum in the evolution of diagnostic tools, new therapies, and prevention strategies for Alzheimer's disease and other associated dementias.
Great. Well, people all over the world this summer were enthralled with the FIFA World Cup, and soccer made it into the AAIC scientific program. One new study presented at the conference, which has not yet been peer-reviewed or published, compared the brains of middle-aged former elite soccer players with controls who did not have a history of repetitive head impacts. What differences did the researchers find between the former soccer players and the controls?
Yeah, the World Cup is really an international phenomenon, and it was definitely very visible at the AAIC conference with people wearing their national colors with a lot of pride. It was a lot of fun. But unfortunately, we're learning that what is referred to often as the beautiful game may have a dark side. We know that people who play soccer can be exposed to repetitive head impacts, either from heading the ball or from collisions that occur during play. And there are more and more data to suggest that those head impacts can have implications later in life in terms of neurologic and psychiatric symptoms. So this study investigated about 140 middle-aged professional soccer players from the United Kingdom. They were in their mid-40s on average. Most were male, though they included a number of female soccer players, which is great. And they compared their responses on questionnaires, cognitive testing, and brain MRIs to controls who did not have a repetitive head impact history. And what they found was that the professional soccer players complained at a higher rate about psychiatric symptoms like depression and anxiety, and also had a higher rate of subjective complaints about what we typically call executive functions. These are cognitive tasks that require planning, focusing, things like that. They did not find differences on performance on objective cognitive tests, though there were trends where the soccer players performed a little bit worse than the controls. And this really conforms to my clinical experience, which is that people that have been exposed to these head impacts very often have complaints, they're noticing things, but we're not always able to capture them with objective tests, especially early on, like in midlife. What was also concerning was that when they compared brain volumes on MRI scans, they found lower volumes in frontal regions, in the thalamus, regions that are really important for regulating emotions, for performing some of these executive functions, and in the soccer players compared to the controls. So in addition to the higher rate of subjective complaints, there were also some objective signal of changes in brain structure that could explain some of the symptoms and potentially foreshadow future problems in terms of neurologic outcomes as these individuals get older.
Are people talking about soccer players wearing any kind of head protection? Baseball is my sport, so I'm not that knowledgeable about soccer, and so I don't know if that's...
Some goalies wear helmets. Goalies are really exposed. They can be exposed to collisions with other players or with the goal itself, actually, the goalposts. Outfield players generally do not wear helmets, but there have been some changes that have been instituted. For example, here in the UK, in youth soccer, heading is not allowed until the age of 12. When those younger kids are heading the ball, their neck musculature isn't as developed, and so they can be even more prone to the effects of impacts on the brain. And so you're starting to see more recognition of this as a safety issue for players, and you're starting to see some rule changes, particularly in youth soccer. But I think these are still early days in terms of understanding the risks and potential mitigating strategies.
Yeah. Well, experiencing repetitive head impacts is known to be the main risk factor for chronic traumatic encephalopathy, or CTE, in American football players. And recently, the family of former Dallas Cowboys defensive end Marshawn Kneeland announced that a post-mortem analysis showed that the 24-year-old who died by suicide this past November had CTE. And what if CTE could be identified before death? Gil, you're the co-author of a study presented at AAIC and simultaneously published in JAMA Network Open about the use of a plasma blood test for phosphorylated tau 217, or p-tau 217, as an indicator of CTE and a biomarker of Alzheimer's disease in former American football players. The two hallmarks of Alzheimer's disease are beta amyloid plaque and pathological tau proteins that clump together into neurofibrillary tangles. P-tau 217 is strongly associated with beta amyloid levels. What did your study find?
Yeah, this was really a team effort as part of a multi-site study called the Diagnose CTE Study. It's led by Dr. Michael Alasko at Boston University, and this is a study that's following former college and professional American football players to try to identify risk factors, resilience factors, and importantly, develop biomarkers that can allow us to detect CTE in living people. Now, the tau protein, as you said, is fundamental in Alzheimer's disease, but it actually is also the main protein that accumulates in CTE. And so in this study, we tested whether an emerging plasma test that has shown very high sensitivity and specificity for detecting Alzheimer's changes could also detect CTE in living people. We looked at about 180 former football players compared to 50 controls and measured their concentrations of p-tau 217 in the blood. What we found was that the concentration of this protein was slightly higher in the football players compared to the controls, but this was mainly driven by football players that also had beta amyloid, the protein that forms plaque in Alzheimer's disease. The p-tau 217 levels showed good correlation with measures of beta amyloid on PET scans or spinal fluid, but not with metrics that we would associate with CTE, like neurologic or psychiatric symptoms or exposure to previous repetitive head impacts. And so the take-home message from the study is that p-tau 217 may be useful in this population for detecting people that have Alzheimer's co-pathology. We know that repetitive head impacts are a risk factor not only for CTE, but also for Alzheimer's disease, Parkinson, ALS, other neurodegenerative diseases. This blood test may be useful in identifying players who have Alzheimer's and amyloid, but is not probably going to be useful for detecting CTE. That said, there are a lot of other potential tau markers that are being looked at in blood, and I'm cautiously optimistic that before too long, we will identify a biomarker that can help us detect the tau protein in CTE as well as we can detect it in Alzheimer's disease.
People with CTE, are they also at a higher risk for Alzheimer?
Well, football players have a really high exposure to repetitive head impact. So it's true that people who've had previous mild traumatic brain injury, one or two concussions, are at higher risk for Alzheimer's, but this is a really different exposure. This is being hit in the head repeatedly, thousands, if not tens of thousands of times over many years. And what the autopsy studies have shown is that these football players, in addition to CTE, do develop Alzheimer changes, and they develop them about 10 years earlier compared to people who don't have that high head impact exposure. And so while we associate CTE with football specifically, because with very few exceptions, everyone who has been found to have CTE has had a high exposure to repetitive head impacts, we're learning more and more that these impacts can also increase the risk of developing other neurodegenerative diseases. Multiple pathologies contributing to neurologic decline is going to be a real issue in these football players, as it is in older adults in general who develop dementia. So it is useful to have a way of distinguishing those who also have Alzheimer's and maybe candidates for Alzheimer's related treatments. but we still really, really need that biomarker that's specific for CTE as well.
Interesting. Another study presented at AAIC, this one published simultaneously in JAMA, found that a p-tau217 blood test might help predict which asymptomatic older adults had the greatest risk over 10 years of developing cognitive impairment due to Alzheimer's disease. LabCorp and Quest offer a p-tau217 blood test. Should cognitively normal people think about getting tested?
So this is a really important large study that looked at about 2,700 cognitively normal older adults who were followed in 6 different research studies and had this protein p-tau217 measured actually in a number of different ways. They were able to harmonize the measurements, and they separated people into 4 groups depending on their p-tau217 levels: a low group, an intermediate group, a high, and a very high group. And then these individuals were all followed longitudinally and over an average of about 5.5 years. Some were even followed longer, up to 13 years. And what they found was that those individuals who had high or very high p-tau217 levels at baseline developed cognitive impairment at higher rates compared to those who had lower levels. In fact, about 24% of people who had a high level and 38% of people who had a very high level had developed clinically significant impairment within 5 years. So this is very useful in helping us understand the natural history of Alzheimer's disease, the prognostic value of p-tau217, and I think the immediate use is going to be in the design of research studies or clinical trials that are specifically trying to understand the risk of developing cognitive impairment, and clinical trials specifically that are trying to enrich their participants in people who are at higher risk of developing cognitive impairment to see if different interventions, pharmacological or non-pharmacological interventions, can delay or mitigate the risk of developing cognitive impairment. In terms of use in real world, I would say it is really very premature to recommend that cognitively normal older adults get these blood tests, even though they are currently available. There's actually a clinical practice guideline around these blood tests that was updated at this meeting and continues to recommend against obtaining plasma p-tau217 and other biomarkers of Alzheimer's disease in cognitively normal people outside of research studies and clinical trials. There are a number of reasons for this. I think these results are very instructive at the group level, but the precision isn't there yet to make an individual risk for an individual person. And a really important caveat is that the participants in these studies, by and large, were highly selected, highly educated, high socioeconomic status, had relatively limited racial and ethnic diversity. And so the results of the study may actually not generalize very well into a real world population. And so currently, we do not recommend that people obtain these blood tests. That may change as we develop models that are more precise for individual person risk, and especially if we have pharmacological interventions in people who have early changes of Alzheimer in the brain but don't yet have symptoms that can prevent or delay cognitive decline. And there are a number of important clinical trials that are ongoing right now that are testing whether the approved monoclonal antibodies that target amyloid plaques can prevent or delay cognitive decline in cognitively normal older adults who have biomarker evidence of Alzheimer's disease.
The lifestyle interventions are things, everybody should be doing. So
exactly. And that's what I recommend, if people are worried about their risk of Alzheimer because of a family history, or they may know they have a genetic predisposition, or they're just worried in general, my recommendation right now is really to maximize the lifestyle changes and vascular risk factor monitoring and treatment and interventions that we know can prevent dementia or cognitive impairment and don't require a drug necessarily, but are things that are in general healthy for everyone to do, and especially with increasing evidence can delay the onset of Alzheimer or cognitive impairment.
On the other end of the spectrum from asymptomatic individuals are the people with Alzheimer's disease or other dementias at the end of life. About half of these patients experience significant agitation, Benzodiazepines, opioids, and antipsychotics are often prescribed off-label for these patients, but those drugs aren't always effective and carry significant risks. A new study presented at AAIC, which hasn't yet been peer-reviewed or published, found that a combination of CBD and THC, the two primary compounds of the cannabis plant, rapidly reduced agitation. How was the treatment administered, and did it have any serious drawbacks?
Yeah, this is a really important unmet need. It's incredibly distressing for patients and family members. And so this study enrolled about 120 individuals with dementia who were either enrolled in hospice or eligible for hospice based on their frailty and other criteria, and had agitation, and randomized them to receive a study-specific compound that included 2 milligrams of THC and 100 milligrams of CBD, or a placebo. So this was really a drug that was compounded specifically for this study. It's not something that is available to patients at this time. And what they found in the study was that there was a significant benefit on a scale that measures agitation for receiving the cannabis products compared to control. That benefit persisted at 12 weeks. In fact, 84% of people were rated to be improved by the clinicians who received the active drug versus only about 30% of the placebo group. So there was a real benefit here. In terms of adverse events in the study, there was actually a higher number of deaths in the people who received active drug compared to placebo, 8 deaths in the active arm compared to 3 in the placebo arm, and this was statistically significant. None of the causes of death seem to be directly related to the drug product, but nevertheless, this is something we will need to follow and require some caution as we think about potentially translating these results into clinical practice.
You know, Gil, and as you mentioned, this treatment is experimental. It's not available outside a clinical trial like this. But there are all these widely sold, unregulated cannabis edibles, with THC and CBD. Does this study at all suggest that people should try these edibles for treating agitation in patients with Alzheimer's disease at the end of life?
You know, Rita, I really would not recommend generalizing the results of this study to other cannabis products that are available, especially unregulated products. It may be really important that these are really purified forms of the active compounds and don't include some of the other ingredients that can be present in cannabis products. The specific dosing matters, and there are more regulated medical marijuana products. If you are going to try this, do it with a physician who has experience with more regulated, medically available products.
2 anti-amyloid monoclonal antibodies are on the U.S. market to treat Alzheimer's disease, and now an anti-tau drug called diranersen is entering a phase 3 trial. Its maker, Biogen, issued a press release about phase 2 results in May and presented more details about them at the AAIC, although the research has not yet been peer-reviewed and published. Tell us about the anti-tau therapy and the phase 2 trial, which did not meet its primary endpoint.
Yeah, this is a program that I'm actually very excited about. Diranersen is an antisense oligonucleotide, or ASO, that binds to the mRNA of the tau protein and degrades it. So basically, the idea is that it's reducing the translation of tau protein, the production of tau protein, and in transgenic models that are genetically engineered to develop Alzheimer's changes, reducing tau expression actually had really profound benefits in terms of the behavior of the animals and in terms of a lot of the other elements of the disease phenotype. This drug showed a lot of promise in phase 1 in terms of target engagement, reducing tau expression in CSF and on PET scans. And so this presentation was about the phase 2 CELIA study, which was a dose discovery study conducted in about 400 patients with early stage Alzheimer's disease, either mild cognitive impairment or mild dementia, confirmation of Alzheimer's based on spinal fluid or PET scans. And these individuals were randomized to either placebo, low dose, mid dose, or high dose of the ASO. The primary endpoint that they were looking for was a dose response on a clinical outcome called the Clinical Dementia Rating Sum of Boxes, or CDR-SB. This is a very popular outcome measure for Alzheimer's clinical trials that includes important elements of both cognition and daily function. So they were hoping to find a dose effect in the sense that higher dose would show greater benefit compared to placebo. That was not the case. What they found was that though all doses showed a benefit compared to placebo, it was actually the low dose that had the greatest benefit, about 26% of slowing on change on the CDR-SB, whereas the changes with the mid and high dose were 14 and 9% respectively. They were able to also replicate the biomarker results from the phase 1. They showed up to 60% lowering of tau concentrations in spinal fluid, and again showed a remarkable response on PET scans where tangles that had already formed and were being imaged by PET at baseline seemed to disappear or at least reduce in intensity, which is really remarkable. We thought the tangles were there to stay, but these results suggest that if you lower tau, they may be more dynamic. Maybe we can actually eliminate some of the tangles. The drug did have some adverse effects. Some of those were just related to the lumbar puncture. I should have mentioned that this is given intrathecal or through a lumbar puncture into the spinal fluid. So there was pain and some post-lumbar puncture symptoms like headache in all participants. But one adverse effect that was interesting was a confusional state that occurred in people who received the drug. And here there was a dose response. The higher the dose of the drug, the more common this confusional state was. And it's something they really didn't present a lot of details on, but is going to be important to try to understand.
If the U.S. Food and Drug Administration were to approve an anti-tau drug, how would clinicians determine whether patients interested in so-called disease-modifying therapies should try the anti-amyloid monoclonal antibodies or the anti-tau drug?
Yeah, that was, other than the World Cup, the big area of conversation at the meeting was trying to make sense of these results. I think we're still learning a lot about tau as a therapeutic target. Tau has a lot of important normal functions. It stabilizes the microtubules in neurons or the cytoskeleton. It has some important cell signaling functions. And so it may be that you can lower tau too much and that may have negative consequences. That if you lower tau a little bit, you can have benefits on cognitive changes and on clinical outcomes in Alzheimer's disease. But if you lower it too much, you might start to interfere with some of the normal functions, and that may actually have a deleterious effect. Now, this is speculation. We don't fully understand it. Hopefully, they'll be able to dig into the data a little bit and understand it better. But it is a curious finding in the study.
Is diranersen administered as frequently as the anti-amyloid monoclonal antibodies, lecanemab and donanemab, which are infused?
Lecanemab is every 2 weeks, donanemab is monthly.
What about diranersen?
In the trial, people received a lumbar puncture every 12 weeks, but the treatment itself was every 24 weeks in the low dose and mid dose group, and every 12 weeks in the high dose group. So the active drug delivery in most of the doses, including the low dose, which showed the most benefit, was just every 6 months. So it doesn't mitigate the problem of intrathecal infusion of drug, but it's not every month, fortunately, or every 2 weeks. It seems like every 6 months might be enough to really have a significant effect. I think many of us believe that the future of treating Alzheimer's is going to require combination therapies, therapies that target different elements of the biology of the disease: amyloid, tau, inflammation, vascular dysfunction, brain metabolism, synaptic function, neurotransmitters. I mean, there's so many different things that are going on in Alzheimer's disease. It's quite a complex pathophysiology. And people are now developing clinical trials that are testing combinations of therapies. At UCSF, we just randomized the first patient for an NIH-funded study called the Alzheimer Tau Platform, led by my colleague, Dr. Adam Boxer. And this clinical trial is testing whether donanemab, an anti-amyloid treatment, an anti-tau vaccine, or a combination of the two can have a benefit, specifically asking the question, is there a synergistic benefit to treating both of these proteins at the same time? I do think that more and more we're going to see combination trials from the pharma side. But yeah, the first one is NIH funded, and it just highlights the tremendous importance of continued federal funding of research in the United States. And it really has made a huge difference in this field.
Going back to talking about lifestyle interventions, a year ago, in conjunction with its presentation at the 2025 AAIC meeting, JAMA published the U.S. Pointer randomized clinical trial, which compared the benefits of a structured, high-intensity lifestyle intervention with those of an unstructured, self-guided lifestyle intervention among older adults at risk of cognitive decline and dementia. Both interventions encouraged increased physical and cognitive activity, a healthy diet, social engagement, and cardiovascular health monitoring. But the structured intervention was found to have a statistically significant greater benefit on global cognition. At this year's AAIC, and simultaneously in The Lancet, researchers reported findings that build on and reinforce the U.S. Pointer results, except this time the lifestyle interventions were culturally adapted for 11 Latin America countries. Both the U.S. and Latin America studies are based on the original Finnish finger trial, which showed that lifestyle interventions may help protect cognitive function in older adults at risk for decline. What were some of the cultural modifications in the Latin American fingers compared with U.S. Pointer?
Yeah, Rita, this is really interesting. How do you implement lifestyle modifications in a culturally competent way? And of course, we think that a lot of the social determinants of health and medical risk factors that could be addressed potentially by these modifiable risk factors have a higher prevalence in low to middle income countries, including many of the countries that were part of this trial. They did something that I thought was really clever. They had some elements of the protocol that were fixed, and that's things like the inclusion criteria, their targets for exercise intensity or specific food groups, or the cognitive tests and the clinical outcomes they were looking at. But they were also flexible about the specific changes that were made to achieve those goals. So, for example, when you're thinking about nutrition, they allowed each country to figure out a diet that made sense to them. In the Finnish study and the Pointer trial, this was a MIND diet, which is a version of the Mediterranean and DASH diet. But in some countries, elements of that diet may not make sense. For example, saying fish are a key element in the diet in a landlocked country that doesn't have access to fish, or requiring expensive berries in a country where tropical fruit may do just as well. And so they allowed each country to tailor the diet, the exercise component, the physical activity component. So, for example, some countries chose to include salsa or tango or group exercise outdoors, which are part of the local culture. They also had a brain training component that included digital cognitive brain games. And in some of those countries, individuals that were recruited didn't have a lot of familiarity with digital devices and needed extra attention or help there. They also culturally tailored the approaches and strategies they used to sustain engagement and ensure compliance with this really intense intervention that they did. And so by combining fixed elements of the study, but also being culturally flexible around the implementation, they were able to show that it is feasible to do this in a variety of different cultures, in a variety of different countries, and in a low to middle income country where resources may be more limited. So I think that's a remarkable achievement. The effect size was actually significantly larger in this trial compared to the Pointer trial in the US, for example. They also found effects on memory that were not found in the Pointer trial. And so they were able to not only replicate, but actually extend some of the findings and really show reproducibility across different cultures. So that was a great trial to see.
Well, Gil, thank you so much for talking with us. This has been really interesting.
Yeah, thank you so much. It's as I said in the beginning, a really exciting time in the field, and hopefully the listeners found some of these advances interesting.
Well, I'm Rita Rubin of JAMA Medical News, and I've been speaking with JAMA Associate Editor Dr. Gil Rabinovici about research highlights from the recent Alzheimer's Association International Conference. You can find a link to the articles in this episode's description. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com or search for JAMA Network wherever you get your podcasts. This episode was produced by Daniel Morrow at the JAMA Network. Thanks for listening.